Hey there, people. Welcome back to the Biology Guru. I'm Jay, and in this video series, we'll be talking about proteins. Proteins are the molecular mechanics within our cells, tasked with a variety of functions essential for life. They catalyze reaction, replicate DNA, respond to cellular signals, and much more. Each protein's function is determined by its unique structure. We'll be looking at how proteins are made and how their structures dictate their roles in cell. We'll also cover protein synthesis, where cells read genetic information to build these complex structures. Throughout this series, we'll explore how understanding proteins at the molecular level is crucial for biotechnology and medical advancement. By manipulating proteins, scientists develop new therapies and improve health outcomes. So, stay tuned as we unravel the complexity of proteins and their profound impact on life. Proteins, from a chemical perspective, are the most structurally complex and functionally sophisticated molecules we know. This complexity isn't surprising once we consider that the structure and chemistry of each protein have been meticulously refined through an extensive evolutionary process. Theoretical work by population geneticists shows us that even a small selective advantage can lead to a significant evolutionary change. A slight alteration in a protein sequence can become predominant in the population over time, showcasing the evolutionary adaptability of protein. In this video, we'll specifically talk about how the specific placements of amino acids in a protein sequence dictates its three-dimensional structure. This structure is crucial because it determines how a protein functions within the cell. At later discussions, we'll connect this structural knowledge at the atomic level to understand how the precise shape of each protein molecule influences its specific function. In proteins, the structure of amino acid determines everything about its structure and functions. There are about 20 different types of amino acids that can be part of a protein, each encoded directly by an organism's DNA and each with its unique chemical properties. Each protein is essentially a long unbranched chain of amino acids linked together by covalent peptide bonds. This forms what we call a polypeptide, and proteins are sometimes referred to as polypeptides because of this structure. The formation of a peptide bond is critical process. This covalent bond is established when a carbon atom from the carboxyl group of one amino acid shares electron with the nitrogen atom from the amino group of another amino acid. This reaction is type of condensation where a molecule of water is released as a byproduct. The core structure of the protein or the polypeptide backbone supports a variety of side chains that protrude from it. These side chains are parts of the amino acids not involved in forming peptide bonds, and they are what uh, give each amino acids its unique characteristics. In this figure, we are looking at a two-dimensional diagram showing a segment of polypeptide backbone along with its attached side chain. Each protein is unique due to variation in the sequence and the number of amino acid it contains. It's the diversity of these side chains that gives each protein its distinct characteristics. In this diagram, the two ends of a polypeptide chain are different. The end with the free amino group is known as the amino terminus or N terminus, while the end with the free carboxyl group is referred to as the carboxyl terminus or C terminus. For simplicity, many illustrations might label these groups as NH2 or COOH, even though in a physiological pH they are usually ionized. The sequence of amino acid in a protein is always written from the N terminus to the C terminus, read from left to right. These side chains can be nonpolar and hydrophobic, meaning they avoid water, or they can carry positive or negative charges, influencing how they interact with other molecules. Some even form covalent bonds readily. Each amino acid is represented by both a three-letter abbreviation and a single letter code. In the classification of amino acid side chains, you will find an equal number of polar and non-polar types. However, it is important to note that However, it is important to note that some side chains categorized as polar may exhibit non-polar characteristics due to their size. Examples of these amino acids include threonine, tyrosine, arginine, and lysine. Atoms can be thought of as hard spheres with a definite van der Waals radius. This concept, along with the constraints on bond angles in a polypeptide chain, severely limits the three-dimensional arrangements or conformations of proteins. These limitations are due to steric restrictions such as the delocalization of electrons in the peptide bond that forces the linkage to remain planar. Despite these restrictions, a protein's long flexible chain can fold in myriad number of ways. Each amino acid contributes three bonds, in this figure highlighted as red, to the backbone of the polypeptide chain. The peptide bond itself has a partial double bond character, making it planar and restricting free rotation around it. In contrast, rotation is possible around the C alpha C bond with the angle of rotation known as psi, and around the N C alpha bond with the angle of rotation known as phi. The folding of a protein is influenced by sets of weak non-covalent bonds between different parts of the polypeptide chains. These bonds include hydrogen bonds, electrostatic bonds, and van der Waals attractions. Through individual non-covalent bonds are much weaker than covalent bonds, their collective strength in large numbers can stabilize the folded shape of a protein. Another critical factor in protein folding is the hydrophobic clustering force. Non-polar side chains like those of phenylalanine and leucine tend to cluster away from water, minimizing their disruptive impact on water's hydrogen bonded network. Meanwhile, polar side chains are generally found on the exterior of the protein, where they can interact with water and other polar molecules. Any polar amino acid buried within the protein usually forms hydrogen bonds with each other or the backbone, ensuring structural stability and functionality. In this illustration, we can see that polar amino acid side chains are positioned on the external surface of the protein. This arrangement allows them to interact freely with water, while the non-polar amino acid side chains are sequestered within the interior of the protein, forming a dense hydrophobic core that is shielded from water contact. Proteins naturally fold into the conformation that minimizes their free energy, driven by the specific sequence of amino acids in the chain. This lowest energy state is generally the most stable three-dimensional structure of the protein. Biologists have demonstrated this process by studying proteins in a test tube. When treated with solvents that disrupt non-covalent interactions, proteins unfold or denature, losing their natural shape. Remarkably, when the denaturing solvent is removed, many proteins spontaneously refold or renature back to their original conformation. This demonstrates that the amino acid sequence itself contains all the information necessary for specifying the three-dimensional structure of a protein. Although proteins can fold correctly on their own, molecular chaperones often assist in this process. These special proteins bind to partially folded polypeptide chains, guiding them along the most energetically favorable folding pathways. In the crowded environment of the cytoplasm, chaperones are crucial for preventing newly synthesized protein from forming aggregates by ensuring that the hydrophobic regions do not stick together. Ultimately, the final 3D shape of a protein is determined by its amino acid sequence, with chaperones making the folding process more reliable and efficient. In the study of protein, we often see two regular folding patterns, the alpha helix and beta sheet. The alpha helix was first discovered in alpha keratin, the protein that forms our hair filament. It forms a rigid cylinder with hydrogen bonds linking every fourth peptide bond, creating a helix with a turn every 3.6 amino acids. Regions of alpha helix are abundant in membrane proteins, like transport proteins and receptors. In transmembrane proteins, the alpha helix often spans the lipid bilayer with hydrophobic side chains facing outwards to interact with the lipid environment. In some proteins, alpha helices wrap around each other to form a stable structure known as coiled coil. This occurs when non-polar side chains are oriented inwards allowing multiple helices to twist around each other. In this conformation, the amino group of every peptide bond forms a hydrogen bond with the carboxyl group of another peptide bond, located 4 residuals earlier along this chain. Notice in this diagram, all the amino groups are oriented upwards while the all carboxyl groups point downwards. This orientation creates a polarity in the helix, with the C terminus having a partial negative charge and the N terminus having a partial positive charge. In this sequence, amino acids at position A and D are situated close together on the surface of the helix, creating a stripe that spirals around the alpha helix. Proteins that form coiled coils typically have non-polar amino acids at this A and D position. As shown in the middle diagram, this arrangement allows two alpha helices to wrap around each other. The nonpolar side chains from one helix interact with those from the other, stabilizing the structure through hydrophobic interaction. The beta sheet discovered in fibroin from silk forms when polypeptide chains align side by side. These sheets can be parallel or antiparallel, creating a very rigid structure held together by hydrogen bonds. Here, adjacent polypeptide chains run in opposite direction, known as antiparallel orientation. Hydrogen bonds between peptide bonds in different strands stabilize the structure of the beta sheet. Amino acid side chains in each strand alternate between projecting above and below the plan of the sheet. Conventionally, arrows are used to represent beta sheets, with the arrowheads pointing towards the C terminus. Beta sheets are common in the core regions of many proteins, providing structural stability. The repetitive hydrogen bonding pattern results in a highly stable and rigid structure. This folding pattern, alpha helices and beta sheets, are fundamental to protein structure, demonstrating how specific sequences and interaction lead to the complex forms and functions of proteins. To fully understand the protein structure, scientists define four levels of organization that build up the complete architecture of a protein. The first level is the primary structure, which is the unique sequence of amino acid in the protein, determined by the gene encoding that specific protein. The next level is the secondary structure, which includes the alpha helices and beta sheets formed by the polypeptide chain. This structure arises from hydrogen bonding between the amino and carboxyl groups in the polypeptide backbone. The tertiary structure refers to the full 3D organization of the polypeptide chain, incorporating alpha helices, beta sheets, and the various twists and turns that occur between the N and C termini. Finally, the quaternary structure applies to the protein that are composed of more than one polypeptide chain, for example, hemoglobin. This structure describes the complete conformation of the protein complex. Visualizing this complex structure is challenging due to the thousands of atoms that are involved and their precise orientation through the covalent and non-covalent bonds. Researchers often use computer-based 3D displays to aid in understanding this complex architecture. In this photo, let's look at the four commonly used representations to describe the structure of protein. This structure is of SH2 domain, which consists of a string of around 100 amino acids and is part of many proteins. Uh, the figure A represents polypeptide backbone model. This model illustrates the backbone of the polypeptide chain, allowing us to see the overall path of the protein structure. The figure B shows the ribbon model. This model provides more detailed view, highlighting the secondary structure such as alpha helices and beta sheets. This model makes it easier to understand the folding and organization within the protein. The figure C shows the wire model. This representation includes the amino acid side chains, providing insights into the specific interactions and fundamental groups within the protein. It's useful for visualizing how side chains contribute to the protein's function and stability. Um, the space filling model in the figure 4, the space the space filling model in diagram 4 shows the protein as a solid object with each atom represented by a sphere. This model gives a realistic view of the protein's size and shape and how it might interact with other molecules. Proteins ranging from 50 to 2000 amino acids long often consists of smaller units called domains. A domain is a structural unit that folds independently, formed from about 40 to 300 continuous amino acids, and acts as modular building block for larger proteins. This figure represents a ribbon model of three different organized protein domains. This example illustrates that the core of a domain can be constructed from alpha helices, beta sheets, or a combination of both. For example, the first figure shows the cytochrome B562. This single domain protein is involved in electron transport in mitochondria. This protein is almost entirely consists of alpha helices. Uh, the second protein is the NAD binding domain of lactate dehydrogenase. This enzyme domain binds NAD and is composed of mix of alpha helices and parallel beta sheets. The third figure is of variable domain of an immunoglobulin light chain. This domain of an antibody light chain consists of a sandwich of two antiparallel beta sheets. Different domains within a protein often have distinct functions. For instance, the SRC protein kinase has three domains, SH2 and SH3 for regulatory roles, and a C terminal domain for kinase catalytic activity. This modular structure allows proteins to function as molecular switches, transmitting information throughout the cell. Understanding these fundamental aspects of protein structure is crucial as it forms the basis for much of the cell biology and biochemistry. In our next video, we'll dive deeper into the roles of these proteins within the cell and how their structure enables their diverse function. So, thanks for joining me today on the Biology Guru. Don't forget to like, share, and hit the bell icon for more videos. I'll see you in the next one. And till then, stay curious.